Thank you. Good evening, everybody. So, as our introducer said, we're going to have a look at enzymes today. This is AS revision for Unit 1, and let's have a look at what we're going to cover in this session. So, we're going to review what we already know from GCSE about enzymes, just to remind ourselves and get us up to speed. We're then going to have a look at some of the key terms that we need to be aware of so that we can use them proficiently in our AS exam, and then we're going to have a look at some exam questions on enzymes and also how enzymes behave. Okay, so let's just remind ourselves what we know from GCSE already. So if you remember, you looked at enzymes as part of your GCSE in Year 10 in Unit 1, and we looked at how enzymes are specific. So we looked at the names of different enzymes. If you remember, we looked at carbohydrases, proteases, lipases, all which break down carbohydrates, proteins and fats or lipids respectively. And we had an understanding that those enzymes, so a carbohydrate, cannot break down a protein, for example. So enzymes are specific. We also should remember that all enzymes have an optimum range. This means that enzymes work within a particular range or a particular set of parameters. So, for example, certain enzymes will work within a pH range. So in our small intestine, it's an alkaline condition, so enzymes will work best that work in that optimum alkaline pH range. All enzymes are made from protein, and if we remember, the structure of protein enables those enzymes to do their specific jobs depending on how the protein is folded. Remember, it's amino acids that make up the protein. Enzymes speed up reactions, and the key point here is that these enzymes would normally be carrying out that reaction, but they speed the reaction up and make it faster. And another piece of information that we should remember from GCSE is that enzymes join with a substrate and form an enzyme substrate complex. And we'll look at some of these terms and areas here from GCSE and expand on them throughout this presentation. You should be familiar with this diagram here as well. And what we've got here, I'll just get my highlighter. Here we've got a diagram that shows us what happens in an enzyme substrate complex. And what we have are our substances here that are joined together entering our active site. And if you remember, the active site is the part of the enzyme where those substances go in that we want to, in this case, break down. Not all enzymes break things down. Sometimes enzymes will build up products as well. So here in the middle picture, this is where our enzyme substrate complex has formed. And then finally, in our end picture here, we have our two products that have been made in this process. So this example here could be something like a lipase, so a fat breaking down enzyme, so a lipase working on a lipid molecule and turning it into fatty acids and glycerol here. Okay, so that's what we should be familiar with from GCSE. So let's move on now and look at some of the terms that we need to be aware of for AS level. So it might be an idea, once I've gone through these terms, to just go back and pause the film and think about how you could remember these terms. So think of an active way of revising these terms. So do you want to make flashcards with these definitions on, or would you make a mind map or some other way that suits you, with post-it notes maybe on sort of a big diagram? or draw some pictures to illustrate it, but you think about which way suits you best to learn these key words and terms, because once you've learnt them, you can then apply them to any question that involves this, and obviously knowledge is power in any of these situations. So let's have a look. The active site is a specific area on an enzyme, and it's the active site that is important for the substrate to join into the enzyme to enable the reaction to take place. If you remember from GCSE, sometimes the active site can become distorted when an enzyme is denatured, when there is a change in perhaps temperature or pH away from that optimum. So the active site is a very specific area, and we will look at that later when we look at inhibitors and see how they work. So here we've got our second term, competitive inhibitors. And this is a substance that joins the active site on the enzyme. So it joins the area where we would want our products to join. And it stops that, it stops the substrate from binding, so our products won't be made and won't be released. So it's called a competitive inhibitor. Now, you'll be familiar from GCSE with the enzyme substrate complex, which is what's formed when the substrate binds with the active site. But we've got a different term here that is new to us here at AS level, and it's called the enzyme product complex. And this is what happens in a reaction when the enzymes catalyse the reaction, but before those products leave the active site. So you've got the enzyme substrate complex towards the start of the reaction when the substrate binds with the active site, and then you've got the enzyme product complex nearly at the end of the reaction before those products leave the active site at the end of the reaction. And here we've got two types of reaction to do with enzymes and how they behave: intracellular and extracellular. So intracellular reactions take place inside the cell, and extracellular reactions take place outside of the cell. So again, we've covered quite a few terms already. So at any point you want to pause the film, do so to make notes to help you with your revision. Activation energy, this may be something that you covered at GCSE, but it's the amount or the minimum amount of energy required for a reaction to take place. And there's an exam question later on that I can use to illustrate this in a little bit more detail. And as I mentioned previously, this term denaturing or denaturation. So when an enzyme has been exposed to a change in its optimum, so either a higher temperature or an extreme of pH to what it is normally used to working at, then the structure of the active site is changed, and that means the enzyme substrate complex may no longer form. And the top term here, the lock and key hypothesis, this is something that you should be familiar with from GCSE. I mentioned earlier about the enzyme being specific and how carbohydrases will work on carbohydrates to break them down. This is the idea from that hypothesis, that the enzyme will only fit into a substrate that has a complementary shape to the active site. So the idea that the enzyme and the substrate are matching like a lock and a key, so the key will only fit into a specific type of lock. But there's another hypothesis that has been postulated, and that is the induced fit hypothesis. So as that substrate binds to the active site on the enzyme, the substrate might change slightly and the enzyme might change slightly, just to make sure that we have an ideal fit to make that fit happen. So it's not necessarily a strict lock and key that matches up complementary shapes, but that there's a little bit of movement that takes place with the enzyme and the substrate so that they comfortably fit together. And we'll look at that later on in an exam question to see how that works. Okay, so what are enzymes? What do they do? So we know that enzymes are made from protein, and we know that they speed up the rate of reaction. And it's how those enzymes are formed and how those proteins are formed that enables enzymes to do their job accurately and efficiently. So they speed up the rate of reaction, they lower the activation energy required for a particular reaction. And as I said earlier, it's important to remember that reactions would still take place, albeit very, very, very slowly. So the enzymes speed up that reaction, lowering the energy required, the activation energy required, so that the reaction can take place very quickly. And enzymes are specific. So the active site of an enzyme will only fit a specific substrate. So we mentioned the lock and key, which is complementary, and this idea of the induced fit, where that active site fits around the substrate. So there are two ideas that you need to be reminded of, and we'll look at these later on in an exam question. And there are some examples here. We mentioned earlier the key terms intracellular and extracellular. So enzymes can speed up reactions inside a cell or outside of a cell, depending on the situation. So we've got some examples here that you might want to make a note of. So intracellular, so ATP synthase and DNA helicase are both examples of intracellular reactions that take place inside a cell. Both are very important in respiration and in reproduction. And then extracellular examples are our digestive enzymes that are secreted by cells and carry out digestion outside of a cell within the digestive system. Okay, so this diagram here relates to the things that might affect the rate of reaction, the speed at which an enzyme works. And there are four particular areas here. So we've got enzyme concentration, the temperature, the substrate concentration, and pH. So you might be familiar with temperature and pH. These are areas and factors that you've mentioned and should know from GCSE. But enzyme concentration and substrate concentration are probably new ideas that you've talked about this year at AS level. So let's have a look at how these factors might affect the rate of reaction. So if enzyme concentration increases, what does that mean? What does that mean for a particular reaction? Well, if you think you've got more enzymes, then on each of those enzymes, you will have more active sites. So that means a reaction can go faster. So more enzymes or an increased concentration means we'll have more active sites, which means more enzyme substrate complexes can be formed and therefore more products. If we look at temperature, then temperature affects the rate of reaction. By temperature increasing up to the optimum, then the rate of reaction can also increase. However, after the optimum, we remember that the rate of reaction decreases because the active sites on those enzymes have been denatured, and that means the enzyme substrate complex cannot form. Thirdly, if we look at substrate concentration, we can see that as the concentration of substrates increases, so does the rate of reaction. So the more substrates we have, the more reactions can take place. And then finally, looking at pH, we've mentioned that enzymes like to work within an optimum pH. However, it can cause a problem if there is a change in that pH. So if it becomes more acidic, then that might lead to a decrease in the rate of reaction. And if it becomes more alkaline, then that might mean that there is a decrease in the amount of products that are formed. So it's important for you to remember these 4 factors and to remember how they might affect the rate of reaction. Okay, so we mentioned a little bit about inhibitors, and there are different types of inhibitor. It's a key definition here that you might need to jot down and be aware of. And an inhibitor is a substance which slows down or stops a reaction. And it stops the binding of the substrate to the enzyme. So it stops that reaction from taking place within the active site, the substrate joining to the enzyme. And there are a number of different types of inhibitor that we are going to have a look at. And they can be reversible, which means they can change and the reaction will then take place, or they can be irreversible. So some irreversible example here includes cyanide that binds to the active site, and that then prevents the substrate binding to the enzyme. And a reversible example is when we might have a substance binding to the active site, and it is very weak here. It's not a strong bond at all, and it's not permanent, and that means that it's reversible and the enzyme can be used to increase the rate of reaction. So let's look at a diagram showing us what happens in reversible inhibition. So here's our enzyme. Here, I'll just get my highlighter. Okay, so we've got our enzyme, and here is our inhibitor molecule here. And you will notice the shape of the inhibitor is the same as the shape of the active site on this enzyme. And this is the key part on this particular type of inhibition. So here in the second picture, the inhibitor has gone into the active site and it's fitted in there perfectly. And the problem with that here, we can see it prevents the substrate from binding. And this is the substrate here, which is what we would want to bind in this reaction, but it can't do that because it's been stopped from doing that by this inhibitive substance here. Now, there are competitive inhibitors, and these have a similar structure to the substrate molecule. They bind to the active site and it competes. So that's what we've just seen there. It competes with the substrate for the enzyme. So because if there is a higher concentration then of competitive inhibitors, it means that that reaction is lowered. Okay, so I'll go back to the diagram and we can see here that this is our inhibitor. It's gone into the space there, into the active site, and it has competed. You can see there that the substrate has the same shape, but the inhibitor has gone in there and prevented that reaction from taking place. There are non-competitive inhibitors, and as the name suggests, these molecules don't compete for the active site, so they don't bind at the active site, but they do bind somewhere else on the enzyme, and that prevents the reaction from taking place, and that is called an allosteric site. That means that the enzyme substrate complex does not form. So in non-competitive inhibition, a molecule will find its way into the enzyme. It doesn't bind at the active site, but it does bind somewhere else on the enzyme. And that means that the active site shape is changed and it won't accept the substrate molecule that we want. And so the enzyme substrate complex does not form. And we've got some examples of inhibitors here. So there are three. There's penicillin, and penicillin is used to fight bacterial infections. It's an antibiotic, and it acts as an inhibitor of an enzyme which is called transpeptidase. And this enzyme normally plays an important part in cell wall formation. So penicillin blocks that and stops that from taking place, and it means then that we become healthy after having our bacterial infection. Then secondly, we have another inhibitor which is called ritonavir, and that's an antiretroviral drug used in the treatment of HIV. And it inhibits a particular enzyme called HIV protease, and that would normally be involved in making new particles, new virus particles, and also would help the spread of infection. So because that enzyme is inhibited, it means people who have HIV won't increase anymore in terms of their virus load, and also the infection won't spread anymore. And then finally, our third example is digitalis. And digitalis you might have heard of. It's a chemical that is extracted from foxgloves, and it's used to treat arrhythmia, which is a heart problem. And digitalis is a non-competitive inhibitor. So that means, if we remember and think back, that the non-competitive inhibitor in this case will bind to a different part of the enzyme, to the allosteric site, rather than the active site, and it will stop the substrate joining with the enzyme. And what that does in this case, it binds to the enzyme and stops or restricts the contraction of the heart, the cardiac muscle. So that means that the heart beats at a regular manner that we want it to. Okay, we're going to have a look through some exam questions now. And we can see here there is an example of a graph. And the question is asking us what is represented by X on the graph. So here we have X. So if we look at the graph, we've got energy and time, and then we've got our products and our reactants. So pause this now for a second and have a think what X might be, and then when you are ready, replay the film and we'll have a look at what the answer is. Okay, so we mentioned earlier that the amount of energy needed, and remember we know this is to do with energy here, can change in a reaction. So the amount of energy needed is, in this case, the activation energy. So those two dotted lines on this graph represents the activation energy, and that speeds up the reaction. You can see there along the time scale here. Okay, let's look at this second question. So there's quite a bit of writing here, so we'll go through this and highlight some of the important parts. Okay, so succinate dehydrogenase, let me just highlight that, is an enzyme found in mitochondria and is involved in respiration. The enzyme catalyzes the conversion of succinate into fumarate. Okay. Using your knowledge of enzyme structure, explain why this is the only reaction that succinate dehydrogenase can catalyze. So this question is asking us to think about the specificity and how specific enzymes are. So let's look at it again. So we've got a very specific enzyme found in mitochondria that is used in respiration, and it catalyzes a very specific reaction. So using what knowledge we have of this structure, explain why this is the only reaction that this enzyme can catalyse. So pause the film now, write down your ideas, and then we'll have a look at the answer. Okay, so this is a 2-mark question, and you can see here that's underlined the active site. So it's asking us to think what the active site is of the enzyme, and it has a very specific shape. And the substrate that we want to break down is here. So this succinate we want to break down into the product, which is fumarate. So the active site of the enzyme is very specific, and the substrate has a complementary shape, and therefore it fits into the active site. And very specifically here you can see on the mark scheme that it tells us not to use the word attach. Okay, so we must use the word fit or bond or bind there. Okay, so again, so we've got an enzyme, it's found in mitochondria, it's involved in respiration, it tells us what the enzyme converts and into what product, and then why do we need to know that this is the only reaction there? So it's the active site, has a specific shape, and then the substance that we are breaking down has a complementary shape to that. Okay, now there's quite a bit of information here in this particular question. So we've got 2 theories here that we've mentioned earlier in this session. So let's have a look and see what we can remember. So we've got theory 1, and we've got our substrate here with a very particular shape. We've got an active site on the enzyme, and you can see here the shape of this active site is very different to the substrate. So start thinking now what theory that is out of the 2 that we mentioned earlier. As that substrate joins into the enzyme and the active site, something happens at X, something is formed. And then after that has taken place, we have our products. And you can see here that the active site, and this is important, and we haven't mentioned it yet, but the active site on the enzyme is now still available and ready for us to carry out that reaction again. So that's an important point that we haven't mentioned so far, is that enzymes can be reused again and again. So let's look at theory 2 and think to ourselves what this theory might be here. So as you can see, as we looked at previously, we've got the substrate here with a very specific shape. And if you look at the active site on the enzyme, you can see that that shape is complementary to each other. So the substrate is complementary to the active site on the enzyme. They join together and form X. So think about what that is called. And then at the end of the process, our products here are formed exactly as above. And you can see again that our active site here is available and is ready to take our next reaction. Okay, so let's have a look at what the exam question is asking us now. So theory 1 shows the induced fit hypothesis. What name is given to theory 2? So let's just go back. So it tells us that theory 1 is the induced fit. And if you remember, we mentioned this previously. So we've got our very specific enzyme shape, The active site is not specifically complementary as it is in theory 2, but as that substrate enters the enzyme, the active site and the substrate enable and carry out a little movement to enable that fitting together, and that fitting together to enable then the products to form. So theory 2, we know it's not induced fit, so it must be the lock and key theory. And then our second question, which theory represents the activity of lysozyme? It is theory 1, the induced fit theory. So let's have a look at question B and question C, both 1 mark questions, so it should be straightforward for us to pick up marks on these questions here. So name X as shown in both theories, and then enzymes are biological catalysts. How do they bring about their effect of speeding up a reaction? So let's go back to our diagram. So pause the film for a second now and write down what you think is happening at X. Okay, so let's have a look at our question. So name X as shown in both theories, and that is the enzyme substrate complex. So you should be happy now that that is when the enzyme has joined with the substrate and the enzyme substrate complex has formed. And it's very important, we can see there in the mark scheme, that we don't abbreviate that in any way, and we must write the full names there, that it's the enzyme substrate complex. So no abbreviations at all there, because you will lose marks. So question C is asking us and telling us here that enzymes are biological catalysts, so we know that they are substances that speed up reactions. How do they do that? How do they bring about their effect of speeding up a reaction? So they lower the activation energy that is required for the reaction to take place. So if the activation energy is lowered, that means that the reaction can be speeded up. Okay, so let's have a look at our next question. What characteristic of an enzyme at the end of a reaction is visible in both diagrams? And this is something that I drew your attention to when we looked at the diagrams. So we'll have a look at that in a second, and then we need to think of 3 factors which affect enzyme activity, but not including, so excluding, the presence of inhibitors. And we need 3 factors, and it's just naming those 3 factors. And then finally, distinguish between intracellular and extracellular enzymes. So that's just a case here of describing or distinguishing, making a difference, clear difference between intracellular and extracellular enzymes. Okay, so we'll go back now and look at the diagram, because this is quite a long question, and let's have a look at what characteristic we can see there at the end of the reaction that is visible in both diagrams. So here we are, we've got both diagrams here, and it's this particular part of the diagram that we are interested in here. And in particular, this part here on the enzyme. So what feature of that diagram in both theories here is something that tells us something about this reaction? Okay, so pause the film and have a think, and then we'll have a look at the answers for these 3 questions. Okay, so what characteristic then of an enzyme at the end of reaction is visible in both diagrams? And we can see there what we mentioned earlier, that the active site is unchanged and that means the enzyme can be reused and the enzyme's active site there is ready for another reaction to take place. And 3 factors which affect enzyme activity, but not including here anything to do with inhibitors. So this is referring back to the diagram we looked at earlier, where there were 4 factors that affected enzyme activity. Let's see what you can remember. So there was temperature, very specifically, not to mention heat. So temperature is one of the factors that can affect enzyme activity. Similarly, pH can affect enzyme activity, but again, don't write the word acidity, so don't put that in your answer. The concentration of enzyme, so remember we said that the more concentrated or the higher concentration of enzymes there are, that means there are more active sites available, and that means reactions can occur more quickly. And then finally, we mentioned substrate concentration. And again, you can see there, don't say amount. So don't say the amount of substance or substrate, don't mention the amount of enzymes. It's very important then to be very specific there with our answers. And then finally, distinguishing between intracellular and extracellular enzymes, which is what we mentioned earlier. So intracellular enzymes, and remember we have to get both parts of this answer correct to get the 1 mark. So intracellular enzymes work inside a cell, and extracellular enzymes work outside of the cell. And it's important there to say, or not to mention, inside the body. Okay, so hopefully this has given you a very broad overview of enzymes and how they behave, and a recap from what you should know from GCSE, and then moving up onto AS level. So a reminder then, we've looked at different types of inhibitors, we've looked at the enzyme structure, and it's important to remember enzyme keywords as well. Make sure that you think of suitable revision techniques so that you can apply your knowledge, go through the practice questions again to help you with that. So thank you very much for your time this evening, and hope to see you again soon.